you. It's a real treat to be talking today, and thank you so much, Nikolai, for inviting me and for organizing this. I was really excited about this opportunity because of exactly what Nikolai mentioned. I think that proteomics could be really powerful in the context of this biological problem, and I hope that this opens the door to new ideas and new collaborations. And so today I'm going to be telling you, as Nikolai mentioned, a little bit about our work studying single cell variation and how it gives rise to drug resistance in cancer. And this work was work that I started as a graduate student in Arjun Raj's lab and worked very closely with another graduate student, Ben Emert, and now have continued to work on in my own lab with the two students mentioned here, one of whom, Guillaume, is actually from Nikolai's lab as an undergrad, which is really cool. So that was my other connection to Nikolai. And so with that, I'll just dive right in. And so this is the problem that we've been working on for a while now. This is the problem of drug resistance in melanoma. So this is a field of BRAF V600E mutated melanoma cells, and they've been treated with this drug called vemurafenib that is a BRAF inhibitor. And after a period of 3 to 4 weeks, when you look back at the dish, what you see is that many of the cells have died, many of them stopped growing, but there's a rare subset of cells that continue to proliferate and grow. And this big foci right here is a resistant colony that has grown from those drug resistant cells. And what's really interesting about this problem is that—
I'm sorry to interrupt you, your slides don't seem to be advancing on the Zoom screen.
Really? So are you only on the front slide?
Yeah, we see the introductory. Now we see it.
Okay, what now? What do you see now?
The front slide.
Huh, okay, well, you know what we can do? You guys can just all see, everyone can see my whole slide. There we go. Does that work?
Yeah.
And I can just click along. Okay, yeah, we'll just do that. That works totally fine. Oh, but the movies will be fine. We'll figure it out. So here you're looking, as I was saying, you're looking at a field of melanoma cells, and this big foci is the resistant colony. And if you look a priori before the cells have seen any drug, there's no distinguishing features that tell you which cells are going to be the ones that ultimately give rise to drug resistance. So if we kind of put the two pictures together, you don't know when you look at the cells beforehand which ones are going to give rise to this resistant colony. And this is a rare phenomenon in the dish. And this is very much analogous to what is seen clinically in patients. So patients with BRAF mutant melanomas are often treated with targeted therapy. I'll know that although now there's different targeted therapies and immunotherapies as well. But what has been seen with targeted therapy is while patients initially respond to these drugs, and a large number of the cells are actually killed in the tumors, after a period of months, the cancer comes back. And we think of this as being a really good model, or this is the model that we've been using to study that process. And so we just started with some really simple, basic questions about what was happening and how this variation can give rise to drug resistance. So we actually first started by looking at whether the resistant colonies were coming from one or were they coming from many cells, and how reproducible was the phenomenology itself of drug resistance. And so here I'm going to show you a movie that we made, and this is actually just a small subset of the movie. We made a movie of a whole culture dish over a period of about a month of the cells being treated with drug. And this is an area that we focused on. It's one of a handful of areas like this, where we do actually see resistance come up. And so here you're going to see initially, as I start the video, the cells growing without the drug. They're growing without the drug, and they continue to proliferate. And I'll stop to highlight that there are these two cells right here that are ultimately sister cells. And what's going to end up happening is that one of them is going to, when the drug is applied, it's going to go on to give rise to this resistant colony. Meanwhile, the other one is going to respond differently, and it's going to actually ultimately ball up and die. And so if we play this video forward, the top one goes on, it divides more, and then all of the cells from that top one, oh, that's the same video again, I need to advance. All of the cells from the top one are ultimately going to give rise to the parent. And so, or to the colony. So if we continue here, you can see, so there was a divergence of fate. Like initially this bottom one ultimately, I think it's down here, it's ultimately going to go on to die, but then the two top ones ultimately go on to give this resistant colony. And what this got us thinking about, while it provided no it didn't provide our definitive evidence for this, but what it got us thinking about is different models where resistance can arise. And so we considered a genetic mutation model, and this is what you might think of, or what we considered a genetic mutation model, where ultimately when cells acquire these mutations, they're not able to revert back to normal. So here you have a cell, it can acquire a mutation, and then that mutation does not revert back, but those are those cells, the ones with the particular mutation, that ultimately are drug resistant. But we considered a different model based on some of our videos and some of our other experimental evidence, where you had cells that could fluctuate between two different states. And we call these states non-resistance and pre-resistance. And we refer to it as pre-resistance because this is a state that exists before the cells have seen any drug at all. Once the drug is applied, then they ultimately go on to form resistance. But we think of this as, but this term pre-resistance we use to specifically mean in the context of an untreated population. And actually through the experimental design of Luria Delbruck, the fluctuation analysis that Nikolai mentioned, and that I'm gonna go on to describe quite a bit more, we were able to show that drug resistance could not, or the way that the model that we were using, it could not fit this genetic mutation model. And this is consistent with other findings in the field for melanoma and actually for other cancers, where there's pre-existing heterogeneity that is ultimately reversible in nature, that gives rise to which cells are resistant. And so we've now been looking at this in quite a bit more detail. And so we initially started with saying, what are the differences that are existing in these cells? What is the difference that makes a cell pre-resistant? And our first approach was using bulk RNA sequencing to look at what are the genes that are uniquely expressed in drug resistance. Our idea was that maybe these green ones up here that are different, or in this pre-resistant state, maybe they're already expressing some of the genes that are part of the resistance signature. And if these genes have a functional consequence and are important for the resistance process, maybe there's some pre-existing heterogeneity in those genes that ultimately determines whether the cells can be resistant to the drug. And this could be both markers, but it could also be genes that are functionally important for bypassing the signaling that is inhibited with these targeted therapies. And so we identified from bulk sequencing what are all the genes that come up when cells are treated with vemurafenib, treated with the BRAF inhibitor. But our question wasn't, okay, what genes are turned on a drug resistance? Our question is there a subset of cells that are already expressing those genes in a drug naive state? And so this is when we came and we turned to single cell analysis. And so we used the bulk seq to guide our selection of genes, but then we used single molecule RNA fish to look at individual cells and say, are these individual cells expressing the markers of a drug resistant cell? And so we looked in an untreated population, just an untreated population of the same type of melanoma cells, and we used RNA fish to actually quantify very accurately gene expression in these cells. And so this is an example of one of such images. Here we have a cell that has very high levels of this gene AXL, which is a drug resistance gene. And this is in this population before there's any drug at all. And this was actually a rare finding. When I was first doing these experiments, I was under the microscope, I was like, I can't find them. Are they here? I had to look around. And so really to get these experiments to work and to get this off the ground, what we had to use was large scale imaging. And so what I mean by this is we did RNA fish in these images. Each one of these we're probing with individual probes for a particular gene, and each one of those individual spots is an individual molecule of RNA. But because these are rare, we had to take a huge number of images. And so to give you an idea of scale here, you can look along at the grid, and each one of these is an individual tiled image. And this allowed us to take data sets where we could get anywhere from 5,000 to 15,000 cells per experiment with like a day's worth of imaging. And so that really allowed us to scale up to the numbers to be on the right scale of what drug resistance was looking like. So to give you context for scale, we would do a cell culture disk, and we'd say that drug resistance is somewhere between 1 in 5,000 to 1 in 10,000. It seems to be in terms of the number of colonies, or the number of cells that, or the ratio of cells to colonies that is coming up on the dish. So we knew that the phenomenon we were looking for was ultimately rare. And so what you're, in this image, what you can see is that there is pretty rare expression of this gene AXL. Each one of these dots is an individual cell. So the dark blue indicates really low, oh, I'm sorry, the axis label is hard to see here, but the dark blue indicates very low expression, and the red means very high expression. And so you can see the scattered red cells throughout the dish that are kind of scattered everywhere around the dish. And each one of those has high levels of AXL expression. But it wasn't just AXL that we looked at from the RNA-seq. We actually expanded this up to look at a whole panel of different genes. And so here you can see these are histograms of different genes that we pulled from that resistance signature. And what we find is that many of the genes that are part of the drug resistance gene signature already have some low frequency pre-existing expression. And you can look across here and you can see there's, you know, some very rare outlier cells in each one of them. With the histogram, the dotted line represents a threshold where we call everything above that a high expressing cell. And that's just part of the analysis we used to look at the frequency of these and compare the frequency of these rare cells back to drug resistance. And then importantly, this was, we wanted to know whether this was something, some sort of artifact of cell culture or something that was unique to something about cell culture dishes or something about the design of cell culture. And so we turned to patient-derived xenograft models, and we can find the same type of phenomenon occurring in patient-derived xenograft models. And now my lab is very interested in trying to look at this further in actual patient tissues as well going forward. So that's something that we are working on. But we know that there is this pre-existing rare expression of these genes, but we don't know for sure that these genes or the cells that express these genes indeed are the same ones that ultimately become drug resistant. And the reason for that is because RNA fish is an endpoint measurement. When we do those RNA fish, the experiments, those are in fixed cells. So we can't take those same cells and say, okay, now let's treat with the drug and see if they're resistant. So what we did is we turned to to antibodies and tried to isolate these populations live. So fortunately, some of these marker genes that we had selected were also cell surface receptors. And so we used antibodies to label these rare populations and sort them out. And so here what we did is we took them, we stained with the EGFR antibody, we sort out that, you know, fraction of a % of the population that expresses, that has high levels of EGFR protein. And in this context, we found that the EGFR correlated, the EGFR staining with the antibody correlated very nicely with the RNA fish. And we sort those out, and then we treat with the BRAF inhibitor. And what you're looking at here is just an image of the dish afterwards. And each one of those circled clumps is a resistant colony. And what you can see is that if you pre-enrich based upon EGFR levels, that you get many more resistant colonies compared to a mixed control. And we did this with other markers, including NGFR and AXL, and now we've moved on and are working with other ones in the lab as well. But we can consistently pre-sort out this population and pick out the cells that are ultimately enriching for the resistance phenotype. So one thing that's interesting about the RNA fish data is that each one of these genes has expression at like a frequency of like 1 in 100 or something along that order of magnitude. But that's not the same as the frequency of resistance, which is ultimately rarer. So we asked whether there was a subset of cells that have many of these resistance marker genes and whether those might have more of a relationship to drug resistance. And indeed, through developing methods to do cyclic, iterative rounds of RNA fish, we were able to find that there is a population of cells that is expressing many of the resistance marker genes, indicating that there is some coordination between transcription of a set of them. And that these do occur on a frequency that's closer to that of drug resistance overall. One of the things, though, that always stood out to me in this image was that the fact that these two cells, or that it's two cells that have high expression of each of these genes. And they're sitting right next to each other, and it's both of them that are expressing these. And that was kind of curious, and I at the time assumed, okay, you know, it's a recently divided cell, and it just recently divided, and it diluted out molecules of each one of these genes, and it's just a matter of time until they decorrelate. But ultimately, through some other experiments and through thinking about this more, what I started to think about, or what we started to kind of hone in on, is whether this phenomenon or whether expression of these rare cell states was something that persists through cell division. And if so, how long does it persist? And so let's just look at a model of what this could be like. So if you have rare variation, so some small fraction of your population is different, you could have things that are rare variation that is non-heritable, and it would just be completely random which cells in your dish have it. So if you take your cells and you plated them really sparsely, you let them divide a little bit, each of these clusters just has some random distribution of cells that have it on or don't have it on. But in the case of something that was heritable, it would look different. And it may actually have different implications for how these states or how these rare cell states are regulated. In the context of something heritable, though, if you have a population and you let it go through a number of divisions, what you'll see is that all the heritable ones, the state that is heritable, will have passed it on to all the cells that came from that same parent cell. And so, but however, if you were to look at these just with histograms like we've been doing for our RNA fish experiments, they would look the same. So the two conditions, or the two hypotheses, were indistinguishable from here. So we set out to try to do these kinds of experiments with RNA fish to learn whether there could be something more involved or more coordinated that would lead to this spatial patterning. And so what we did was exactly what I showed in the diagram before, where we plated cells really sparsely on a dish. We let them grow up into clusters, and then we did our RNA fish, which is an in situ assay. So now we can look at single cells, we're getting single cells, but we're also getting their spatial position. And now spatial position can mean a couple things. It can mean proximity, it can mean some degree of relatedness because they're close to each other. It also probably has some intrinsic, they're probably, being close to each other means they're in the same environment as well. So there's probably extrinsic effects that come from the cells being close to each other. But through these experiments, what we were able to see was that the spatial position of high cells showed a clustering effect. And so here what you're looking at is for 3 different genes, you're looking at just the high cells in light blue. And so if you look and you start at NGFR, you can see clusters of NGFR-high cells happening in these clumps. You can see the same thing for AXL, and you can see the same thing for Wnt5a. And so there's a cluster in here that is consistent across all 3 of these that has shown longer lived expression. And so in each of these, because we let them grow up in these clumps and these weren't randomized on the dish, we then honed in on the idea that there was some sort of, whether it be intrinsic or extrinsic, but some sort of coordination or related heritable expression of the states in these cases. And so this got us thinking about a model or a model where you have timescales for variation. And so on one end, we had considered initially a genetic mutation as a possible mechanism of resistance. This would be like a slow biological timescale because ultimately it's not reversible. On the other end, you have things that are fast, and something that's fast could be like a burst in transcription or some little blip, something that's very short-lived. And in the middle is what we think we have here, which is a longer lived gene expression state that is ultimately reversible. And we think this might be important because here we kind of came across this as a model for drug resistance in melanoma. But if you're thinking more broadly about how do you generate variation that has functional consequences for cells, well, you need it to involve many different genes. It probably involves, some sort of epigenetic regulation. And it probably leads to many, bigger changes in the cell in terms of the protein contents and what all the different interactions are that are going on inside the cell. This is probably a more functionally different cell, not just like some little blip in transcription leading to a blip in whatever other downstream processes. And so we think that this medium intermediate timescale might be a place where we have non-genetically encoded, functionally important variation. And so here's just an example of seeing these clusters of high expressing cells in melanoma. Oh wait, now you're in my silenced, my hidden slides. There we go. And so then we set out, and so in the context of melanoma, we knew what genes we were looking for because we'd done all this RNA-seq and we had learned a lot about, what genes are expressed in drug resistance. But the idea came up, could we come up with a way to find these gene expression states de novo in an unbiased fashion? And so this is trying to identify these medium timescales. And we turned to the experimental design of Luria and Delbruck as fluctuation assay. And they had used this fluctuation assay back previously to distinguish Darwinian and Lamarckian processes in the context of bacteria and resistance to phage. But ultimately, their assay itself, and I'm going to go into detail and explain it in just a second, is really a beautiful quantitative way to measure how heritable or how long-lived in terms of cell divisions a particular state is. And I use state extremely vaguely here. So the experimental design and how this works. I'm going to do it in the context of my work instead of the bacteria and phage example, but their experiment was exactly the same, basically. So in the context of variable gene expression, if you have something heritable versus something non-heritable, ultimately you have two different scenarios. And so here, this experiment is only done once, but I'm giving you a heritable, what would happen if you have a heritable thing versus a non-heritable thing side by side, so you can see how it looks different. And so what we do is we take individual cells and we put them in separate wells. And what we're going to do is we're going to allow these individual cells to grow out into full, clones, like isolate individual clones. And we've done this in our previous work for 48 clones, and we're playing with different clone numbers and things like that. But previously we've done this with 48 clones, and that's what the data I'm going to show you looks like. And so in both of these scenarios, you take your individual cells, you put them into separate wells, and ultimately, as they start to grow up, you can see the difference between something that's heritable and something that's not heritable. So the non-heritable thing flickers on and off between different cells, and the heritable thing is passed on whenever a cell turns into that state. So a cell enters the state, and it's passed on through all the related cells. And what you can see is that as these grow up, these two scenarios look really different. In the context of something heritable, you have a bunch of wells in your dish, in your 96 well dish, that have cells that have very little or no expression of a heritable gene, or no expression or nothing from your heritable state. However, in the example of something that's non-heritable and entirely random, it's just flickering on and off in all of the cells at whatever frequency, and it has a random distribution. And what you can do after you have all of these individual wells and individual clones is you can take each one of them and combine it with RNA-seq. So we took all 48 clones and we do RNA-seq on them individually. And what we ultimately get is data like this. I'm going to give an example. So here on the left, we have an example of a heritable gene. We have EGFR. And here you can see the actual raw data. These are actual transcripts per million we got from our experiment, where you can see there's some wells that have super low levels, and there's some cells that have super high levels. And what this tells us is in those that have super high, a cell turned the state on earlier, and a huge number of cells ended up with it because it was heritable. However, if you have a state that's just turning on and off at random, more like something regulated by the cell cycle or something, with fast fluctuating variation, every one of the different clones is going to have a similar level of expression. And so the variance here across all the different clones is an indicator of how heritable that gene is. And because we combined it with RNA-seq and not just, not just measuring one gene at a time, we get this for every gene in the genome. So we get a genome-wide measurement of how heritable a particular gene expression or expression of a particular gene is. And then there's important technical controls that we did along with this. So we just said, how much variation do you get from doing RNA-seq 48 times? And so we did these technical variation controls where we just isolate the same number of cells, but we don't go through the clonal bottleneck. And so the clonal bottleneck is the key feature of this, because that's where you generate ultimately the low number of cells that then can expand up and give you the jackpotting behavior that is seen in the Luria Delbruck experiment. And so we implemented this in our melanoma system, where we knew a lot about resistance. And we started with just saying, do we see that the resistance genes show this increased variation across the clones? Can we find heritable expression in those cell lines? And so here, this is EGFR. This is the data I showed you before. But here is an example of a histogram across all 48 clones. And so the blue here is the histogram from the memory-seq clones. And there's a little rug below it. You can kind of see where all the points are, although it doesn't look great on black. But you can see that across all the clones, we have a very wide variance. This is very, very wide. And then in pink, you can see from those technical controls that I was just describing that it's much more narrow. And this is our indication, or this is how we actually go about quantifying how heritable a particular gene is. So we looked at mini genes from, we assayed the data genome-wide, but we looked at mini genes from drug resistance here on the left. And what we can see is for the highest and the lowest expression levels, we can consistently find this behavior. We can find these wide variances indicating heritable expression of these states. In the right column, though, we have some control genes that we found do not show heritable expression. And we know that because the blue distribution is much more like the pink distribution, whereas in our heritable genes, the blue distribution is really wide. And some of these are selected, like CCNA2, because they're associated with the cell cycle, or MYC is a gene that we have done a lot of RNA fish for. We know it's variable, but we don't think it's heritable. And so it serves as a really nice internal control here as well. And as I mentioned, we get this on a genome-wide scale. So there's a whole subset of genes that we find out of this assay that are showing this long-lived memory expression. And so on the plot on the left with the memory-seq clones, you can see all those. We've labeled them with the blue dots. There's a large number of them. And using the same kind of cutoffs and thresholding methods, you can see very few with our control sample that are coming out. And interestingly, when we implemented this in melanoma, it wasn't just the same genes that we knew from drug resistance. We had many of the genes that we knew from drug resistance, but there were other signatures also coming out of that data. And so there's some that by looking at the correlation structure, we can try to predict whether they're in the same cells as the drug resistant cells or other cells. But where you think we're finding an orthogonal population that's in a separate subset of cells that also shows this behavior that is related to the type 1 interferon signaling pathway. And so we've been trying to think and look into that in more detail, but you can see some of those genes here. There's a huge number of interferon response genes, and there's some subset of these melanoma cells that are having heritable expression of these genes as well. There's other gene sets in here as well, but that one was particularly interesting because it just shows such a strong correlation and such a strong signal. Also, interestingly, we've now implemented this in 4 different cell lines. And one thing that we're seeing is that while a lot of the genes are different, there is a subset of, I think it's like 6 or 7 genes that comes out every time we do this. And this is, wouldn't happen by chance. So there's some subset of genes where, regardless of which cancer model we're in, we continue to pull them out as heritable genes. Another thing that we've been interested in for a while was actually trying to directly visualize these gene expression states. And so Ben Emert from Arjun's lab built this really cool reporter system. And what he did is he actually tagged one of the genes, one of the proteins in EGFR at the endogenous locus, and then made these time-lapse movies so that we could see cells divide and maintain the state. And so if you watch this video, you can see the cells turning it on. And then I think he applies drug in this video midway through. And you can see the drug-resistant ones survive. You can see clusters turning it on, clusters turning it off, drug added, and then ultimately a colony grows out. Although at the end, I think there's some amount of death everywhere. But we did do more quantification of this in the absence of drug, just to show both directly that we had some cells that showed very long-lived expression or long-lived persistence in these states, but we also had cells that would revert back. So we have both have heritable states, and we have reversibility occurring that we can directly visualize with the reporter. And this is just quantification of that as well. But one of the ideas that we've been really interested in is why does this happen, and how does it relate to other phenotypes? Or how does it relate to phenotypes? And so one of the things that we really wanted to do was to go into another cancer system or cancer cell line, cancer model, and say, can we use this method to find rare states that drive drug resistance? And so we went with this breast cancer cell line called MDA-MB231. And what we found with this cell line is that there's a range of doses with Taxol where you can get kind of this intermediate response where most of the cells die, but there's a subpopulation of cells that are surviving. And so we implemented memory-seq in these cell lines. And this is actually looking at the correlation structure, which I show because there was this huge, strong signature that came out all together as correlating really nicely. So we targeted this big, strong signature because we thought this is, these are the cells that are really different. This is the rare variation that exists here. And we found a cell surface antibody for one of these genes. And we did the same kinds of experiments as we did in melanoma. So here we labeled these cells with CA9 and we sorted out different populations based on their CA9 expression. And when we treat with the drug, what we find is that indeed CA9 population, this rare subset, is much more resistant to Taxol than the CA9 low cells. And so we went into a system where we knew nothing, we were completely naive to these, and we de novo discovered a gene marker of, or de novo discovered the state that mediates which cells are more resistant to the drug. And this is just quantification of that, that phenomenon. And so going forward, we're really interested in this idea of gene expression memory, although it could be memory more broadly, memory in the context of the whole cell and the makeup and composition of the cell, and what other phenomenon it may underlie. So we've worked in drug resistance, but we've been thinking more broadly about other cancer phenotypes and also more development phenotypes as well. And just to wrap up, I want to say that one of the cool things, or one of the things I really like about the Luria Delbruck experiment, is that it's compatible with any really experimental assay that you want to do. So I am happy to tell people more about it. You can do anything at the end point. So it gives you a way to make a bulk measurement and learn something about a single cell difference. So if you have a measurement like where you want a single cell difference that is heritable. So you can now make a really high quality bulk measurement, but still learn about single cell differences. And so we've been thinking about, different sequencing assays to implement this with and other, other readouts going forward, but the experimental design is applicable to in many different contexts. We've also been thinking more about what this might mean in normal cells and whether this happens in normal healthy cells as well. And so with that, I should acknowledge, so the work I presented today, a lot of it I started in Arjun's lab and did with a graduate student, Ben Emert, and then Guillaume and Raul have really carried this stuff forward in my lab and continued with a lot of this work as well. And of course, I worked with a bunch of other people in Arjun's lab, and I should acknowledge, funding sources and my postdoc advisor, Junwei, as well. So